part 2 and see as to what happens under meiosis part 2. Notice meiosis part 2 is written with your Roman numeral over here. You should be able to recognize meiosis 2 versus meiosis 1. In meiosis 1, we had only one Roman number, and this is meiosis 2. Now, meiosis 2 begins with what happened just after meiosis 1. Now, remember during the stage which is called as interkinesis, just like that interphase, but interphase was longer in the regular cell, and this is interkinesis because immediately the cell is going to go into a second kind of division. So there is no synthesis of additional DNA or duplication. That's why it's called as interkinesis. Now, meiosis 2 is very similar to mitosis. So the stages of meiosis 2 is going to remind you of mitosis, and pretty much now, since its two sister chromatids are attached, the purpose of meiosis 2 is to separate out the two sister chromatids. Recall that mitosis was the same. We had two sister chromatids and they were separated. Now the difference is when the cell undergoes mitosis, it was a diploid cell. However, when the cell undergoes meiosis 2, it's a haploid cell. So here we have a haploid cell that we will see that it will separate out the two chromatids. So basically the same as mitosis, except cells are haploid and they're not diploid. So let's look at meiosis 2 from these images over here. You can see that in meiosis, actually, I'm trying to think. Did I skip a slide over here? Let's see. Let's go back. Look at the previous slide. No, we did do it. So what we wanted to point you is here you have the independent, the two haploid cells that were coming out. And this is the prophase. It's followed by a metaphase. Why is it metaphase? Again, you have the chromatids that are aligned in the center, which is followed by the telophase. I'm going to write that down. So the first stage is prophase, and this is going to be followed by metaphase. And after metaphase, you will have the anaphase. And in anaphase, you can see the two chromatids are now actually separating. And eventually, since it's going to separate out, and it will eventually give rise to what is called as the telophase. And in the telophase, you can see that you eventually are having each nucleus having one chromatid within that. And notice that each of the chromatids is independently assorted. So you have one long red and one long red over here with the blue segment and one blue in here. So you really don't have two long ones on one nucleus and you don't have two short ones on the other. So each of the two chromatids are totally separated. So you have exactly a short and a long chromosome in each nucleus. And eventually, after telophase, when the membrane will be reformed, the cell will eventually give rise to what are called as the daughter cells. And the daughter cells are now all haploid, and they have a variation of genetic material. They still have the two chromosomes that we began with, but if you notice, if you compare each of the chromosome, if you look at the long ones, this is a long red, and then you have a long red with a blue segment. We have a long blue segment and a blue with a red. So we have all kinds of 4 different combinations that is resulting. Now, if this variation did not take place, then the haploid cell will be identical to the parental cell. So this results in a unique variation in the genetic outlook of species. So keep in mind, meiosis 2 had 2 parts, meiosis 1 and meiosis 2. Meiosis 2 is very similar to mitosis. So this will end this mini lecture, and then we have one more short one to go after this.